Hi, welcome to yet another video on membrane transport. This is the penultimate video. And in this video, we're going to talk about osmosis, which is basically the diffusion of water or the transport of water across the membrane. Okay? It has a special name because there's a lot of special things about it. Almost all cell membranes are permeant to water. I say almost, there are some exceptions, particularly in the kidney. But most of the time, water has no trouble getting across membranes, and it diffuses very freely across. It uses, a lot of the time, it uses a water channel in the membrane, aquaporins, that allow the water to get across very easily. And most cell membranes, again, very highly permeable to water, and this has physiological consequences, and it has consequences that are very important to you as a physician, particularly when you think about things like giving IV fluids, or when you think about organs and like the kidney and how they work, or the GI tract, and how you get things like diarrhea. So water, again, no different than any substance in the body. Water diffuses from high concentration to low concentration, just like sodium, just like glucose, just like everything else. All right. So the difference in osmosis and what's special about it is we don't generally consider water concentration per se. Instead of talking about water concentration, we talk about the concentration of dissolved solutes in water. And as you'll see here, it's basically the same thing. So here I have 2 beakers of water. In beaker 1, I have 1 liter of 100% water. Okay, that's the sort of lightly colored dots here. Those are water molecules. All right, this is all water. I can count the number of molecules, and it's got a given concentration. Okay, now let's take a look at beaker 2 on this side, and now we have our infamous black dots in this beaker, surrounded by water, but we have black dots. Now each one of these beakers has exactly 1 liter in it, okay? But because I have these black dots in here, and because they take up space, if I were to actually count the number of molecules in beaker 1 and count the number of molecules in beaker 2, I would have fewer water molecules here than I have here, because for every one of these black dots, I have 1 less water molecule, okay? So the water concentration in beaker 2 is lower than the water concentration in beaker 1. So we say that beaker 2 has a higher osmolarity than beaker 1, and what that means is it has a lower water concentration than beaker 1. So what does that mean? Well, if I put a membrane between these two, and of course we know water moves across membranes with no problem. I have a high concentration of water on side 1. I have a low concentration on side 2. Water is going to move from side 1 to side 2. And it's going to move until the osmolarity is the same on both sides. So water moves very easily across a membrane, and it's going to move from low osmolarity to high osmolarity, or it's going to move from high concentration of water to low concentration of water, okay? Animal cells cannot stand water gradients. Water gets across membranes, and it's going to flow across membranes until the concentration of water is equal on both sides. So at steady state, if I were to look at the cells of your body, and I were to look at the osmolarity inside those cells, and the osmolarity in the extracellular fluid outside the cells, and the osmolarity in the plasma in your blood, and the osmolarity inside your red blood cells, the osmolarity is the same everywhere, because water will flow across those membranes until the water concentration is equal, and that means the osmolarity has to be equal at steady state in all those spots in your body, okay? So animal cells cannot stand water gradients. If I say the osmolarity of the fluids in your body is 300, then that means the osmolarity inside the cells and outside the cells, everywhere in the body, all 300. Because there can't be water gradients in the body, at least not outside the kidney. Okay? Okay. So Again, the osmolarity of a solution is the concentration of dissolved particles. So you add up all the particles, and that concentration is the osmolarity of the solution. The higher the osmolarity, the lower the water concentration. So if I have 150 millimolar glucose, that means I have 150 milliosmoles per liter in that solution. 150 millimolar glucose particles, 150 milliosmoles, okay, in a liter. If I have 150 millimolar sodium chloride, you guys know from your inorganic chemistry classes in your undergraduate days, you thought that was all long gone. Sodium and chloride dissociate, and they form separate sodium and chloride molecules in solution. So we have 150 millimolar sodium chloride, that's 150 millimolar sodium and 150 millimolar chloride in solution, so it's 300 milliosmoles per liter. 300 milliosmoles of particles per liter. If I have calcium chloride, CaCl2, how many milliosmoles per liter? If this were a lecture, this is where I would ask you and then take great joy in the fact that you were wrong. And then I would reveal the solution and tell you that it is, in fact, 450 milliosmoles per liter. 150 milliosmoles per liter of calcium and 300 milliosmoles per liter of Cl2 of chloride. 450 milliosmoles per liter altogether. You add up all the particles, okay? All right. So when we refer to the osmolarity of a solution, we usually call it isoosmotic, hypoosmotic, or hyperosmotic. And that means that in the case of isoosmotic, that means that your solution has an osmolarity that is the same as a reference solution. So let's say I'm holding a bag of solution that I'm going to put in a patient, okay? And I say that this bag is isoosmotic to the body. That means that the osmolarity of the body solutions, the osmolarity of the plasma, is the same as the osmolarity of the solution in this bag, okay? Hypoosmotic means that the osmolarity is less than your reference solution. If I'm holding a bag, I have the body's osmolarity, I have the plasma osmolarity. If the bag has fewer milliosmoles per liter than the plasma, I say it's hypoosmotic relative to the plasma, okay? And if it's hyperosmotic, I say my bag has more milliosmoles per liter than the number of milliosmoles per liter in my plasma, okay? So it's the osmolarity relative to a reference solution. Ordinarily for us, that's going to be relative to the plasma or relative to the inside of the cells, which physiologically is the same thing, okay? All right. A tonicity is a different term, and it's one that's distinct from osmolarity. Tonicity tells you what a cell is going to do if you drop it into that solution. So if I have something in a bag and I say it's isotonic, that means that if I take a cell and I drop it into that solution, the cell volume will not change, okay? So it's isotonic if the cell volume stays the same if I put a cell in that solution. Cells will swell when placed in a hypotonic solution. So if I take a cell and I drop it into a solution and that cell swells, that means water moves from the solution into the cell and it swells. We say that's hypotonic. Hypotonic solutions cause cells to swell. Hypertonic solutions cause cells to shrink. If I drop the cell into a solution, into a beaker, and water moves out of the cell from inside to outside, that cell shrinks and we call that solution hypertonic, okay? So note that osmolarity is a physical property. I can just count the particles, divide by the number of liters, that tells me what the osmolarity is, okay? It's just like concentration of sodium or sodium chloride or concentrations of chloride in millimoles per liter. I count the number of moles, divide by the liters. That's a physical property of the solution. The same thing with osmolarity. You count the particles, you divide by the liters, that gives you the osmolarity. Tonicity is a biological property of the solution. That tells you what will happen to a cell when you put that cell into that solution, okay? And that's different If the cell swells, we call it hypotonic. If it shrinks, we call it hypertonic, okay? So tonicity is different. It tells you what a cell will do if you put it into that solution. That's different from osmolarity, which is just a physical property where you just count the milliosmoles per liter. And they are not the same, as we're going to find out in a minute. You can have hyperosmotic solutions that are isotonic. Okay, the osmolarity doesn't tell you what's going to happen with tonicity. You can have a hyperosmolar solution and have it still be isotonic. I'll show you how that can happen in a minute, in very, very general terms. Okay, so that brings us to cell volume regulation. Since we're talking about tonicity, we're going to concentrate mostly on osmotic effects. That is, water moves from high concentration to low concentration. That is going to cause a cell to swell if the water is moving into the cell, or shrink if the water is moving out of the cell. And that's what we're going to really concentrate upon, how this will regulate cell volume and cause it to change. You should recognize that this is a very complicated topic, and we're not going to talk a lot about it because it's not really necessary for you to get into the nitty-gritty details. But membrane potential is going to affect cell volume. If I have a membrane potential that changes, that's going to cause particles to move across the membrane, and that's going to cause your cell volume to change. You should recognize that. You should recognize that active transport can have effects on cell volume. You have a sodium-potassium pump, which we just described. That moves 3 sodium out of the cell and 2 potassium in the cell. So every time it turns over, it moves an extra sodium out, okay? So that has an effect on cell volume regulation, as I'm going to talk about in a moment, okay? The constituents of the intracellular and extracellular solution, I think that's obvious, particularly, if you're changing the osmolarity, you're going to have an effect on cell volume. And of course, the membrane permeability is going to have an effect. If you make a membrane more permeant to a substance and that substance moves across the membrane, you can change the osmolarity and you can alter the cell volume. Okay? So, again, you need to recognize that, in spite of all these effects, when water is done moving, you are going to have the same osmolarity inside and outside the cell. Okay, and when you have the same osmolarity inside and outside the cell, that is going to be where your cell volume is going to be at steady state. Okay, so water is going to move until it stops moving. It's going to stop moving when the osmolarity is the same on both sides, and that tells you what the cell volume will be. Okay, permeant molecules, membranes that easily cross the membrane, have no effect on cell volume. Because what happens is, if I have a membrane here and I add molecule 1 to this side, molecule 1 is just going to go from this side to this side, and eventually I'm going to have the same concentration on both sides of the membrane. So that has no effect on osmolarity. Eventually I'll have the same osmolarity. So I haven't affected cell volume. On the other hand, if I have an impermeant molecule, that is going to affect cell volume. So here's my membrane. If I drop a membrane impermeant molecule here, now this molecule can't get across the membrane. So I have a higher osmolarity here than I have here. So if this molecule can't move, water will move instead. And it will move from high concentration to low concentration. And it will do that. Water will move from here to here until the osmolarity on this side increases and the osmolarity on this side drops to the point where the osmolarities are equal again. So membrane impermeant molecules are the ones that cause water to move, and they're the ones that affect cell volume. So let's talk a little bit about how active transport affects all this. Here's my sodium-potassium ATPase. Now at steady state, I'm constantly moving 3 sodiums from inside to outside and 2 potassiums from outside to inside. Now, I have a steady cell volume here, and what that tells me is that despite the fact that my sodium-potassium pump is moving an extra particle out, there are other things in this membrane, other transporters, other channels that are allowing extra particles to come back in, such that the osmolarity at steady state isn't changing. So every time this pump turns over, it moves a particle out, but something else is moving a particle in. So at steady state, the osmolarity is constant, and it's the same here and here. Okay, your red blood cells are doing this right now. Okay, and the cell volume is constant because you've got this constant trade-off of transporters such that the osmolarity is maintained the same on both sides. So now, what happens if I inhibit this pump? And you will do this very frequently as a physician, using something like digitalis to treat heart disease. So I add digitalis, I inhibit the sodium potassium pump. Now, I'm no longer moving this extra sodium out. So what's going to happen is that sodium is going to accumulate inside the cell. And as that happens, the osmolarity increases. Well, if the osmolarity is increasing here, and it becomes greater than the osmolarity on the outside, cells can't stand water gradients. So in response to that, water is going to move from outside to inside. And it's going to do that in order to keep the osmolarity the same here as it is here. Okay? So water moves in response to the pump inhibition, in response to the fact that we're leaving behind the sodium. And as that happens, the cell gets bigger and swells because there's more water in it. Okay? So you get swelling of the cells with digitoxin treatment, and that can cause side effects, as you might expect. So cells swell, okay, if you add a sodium pump inhibitor. Okay, so that is a very easy case where you can see that this isn't just gobbledygook, it's actually relevant in real situations. Here's another situation, we're going to talk about this a lot in the vital fluids block. We'll introduce it here. Here's a capillary. Blood is flowing from here, the arterial side, to the venous side. Now, the capillary wall allows water to get across from the lumen of the capillary into the tissues. Okay, so water crosses this very easily. It goes through the cells, no problem. But these walls are impermeate to plasma proteins, primarily albumin, because they're too big to get across the wall, and the proteins won't go across. So what that means is that I have a higher osmolarity in the blood because of the existence of this albumin and the fact that it can't get across the wall. The osmolarity is higher here than it is here. So we say that the capillary has a high oncotic pressure. It has a special name. And all that means is that water tends to move from the tissues into the capillary because the osmolarity is higher here.
Sorry?
So what we call that reabsorption of fluid, okay? That actually is opposed by what we call filtration. So I said blood is flowing from the arterial side to the venous side. That means the hydrostatic pressure is higher here on the arterial side, close to the arteries, than it is here on the venous side, close to the veins. So I have a pressure of 30 millimeters of mercury there. I have 10 here. Blood flows from high pressure to low pressure. So we have a high hydrostatic pressure. And the hydrostatic pressure is higher in the capillary than it is in the tissues. So not only is blood flowing from arterial side to venous side, water is also flowing from the inside of the capillary to the outside of the capillary into the tissue. Because the pressure is high here and low here, it flows across the wall as well as down along the capillary. And we call this movement from here to here filtration. So we have reabsorption, pulling water in. We have filtration, pushing water out. And you'll notice that the filtration is higher here than it is here because I have a higher hydrostatic pressure here pushing water out than I have here pushing water out. So what happens to water is a balance between those two things, okay? On the arterial side, filtration wins, and we push more water out through filtration than we reabsorb through our osmotic gradient, okay? On the venous side, the hydrostatic pressure is lower, we're not filtering as much fluid, so on balance we reabsorb more fluid, that is more fluid moves down the osmotic gradient than we shove out through filtration. So we have a net movement of water into the capillary on this side, okay? On balance, you move slightly more water out across this entire capillary than you move water in. And so that eventually becomes lymph, okay? And that's where your lymph comes from in your body. All right? If you have a disease state where you're not reabsorbing enough fluid because the osmolarity is too low, let's say there's low protein, or you have a disease state where the hydrostatic pressure is really high, you can upset this balance and you can cause too much water, for instance, to accumulate in the tissues. That causes tissue swelling because they become turgid with fluid, and you get what's called edema. Okay? So we're going to find that happens a lot in cardiovascular diseases. It happens in nutritional problems. It happens in kidney problems. This is going to become very relevant. We're going to be dealing with issues that are related to the osmolarity all up and down the curriculum. So it's best that you get used to it and you make sure that you understand it now, early on. Okay, I'm going to do one last video where we solve some problems related to osmolarity. And I'll expect you to be able to do these problems. They'll be related to some of the problems that you'll do in the computer workshop that we're going to do. And so we'll get on to that. Thank you for your attention.